This lecture covers some of the details on microbial growth. In order to grow, microbes have to replicate the chromosome and then partition it as the cell is elongating. And so some of the constraints that are applied to bacteria are considered in this lecture as well. Most bacterial chromosomes are circular. They have a single origin of replication. The origin of replication is the site at which replication begins in a circular chromosome. The terminus is where it then would stop replicating. And the replisome is the group of proteins needed for DNA synthesis, so the polymerases and helicases and single-strand binding proteins and so forth. DNA replication proceeds in both directions from the origin in prokaryotes, so it's bidirectional. And the origins of replication initiate at one pole, but then move in opposite directions and terminate at a terminus. And so that's pictured here. You see a typical rod-shaped bacterium with its circular chromosome, and then the origin of replication here, so that's where the replisome would assemble. And then you see these replication forks moving bidirectionally, two directions. Here are the terminators, the termination region. And so as these replication forks move around, they bang into the termination region here, and they've basically completed a full round of replication, making a full chromosome. And then, and obviously the termination regions are essential to make sure that replication doesn't go past that. So you get a chromosome and a half or anything like that. You precisely get an extra chromosome for each bacterial daughter cell. The cells can then divide, and then replication can begin anew. And so some of the components that are facilitating this chromosomal partitioning are illustrated here, one of which is MREB. So MREB is an actin homologue. It plays a role in determining cell shape. It kind of forms a spiral inside the cell periphery, as I'll show you, and is essential for chromosome segregation. New origins of replication associate with the MREB tracks. So the origin of replication knows where to set up. And one of the scientific pieces of evidence for this is that if MREB is mutated, chromosomes don't segregate. So we know that it's important. Another way that chromosomes segregate is through mesosomes. These are invaginations of the cytoplasmic membrane. And so the chromosome can associate with these mesosomes. And then as the cell elongates and the chromosome is dividing, they can physically be pulled apart by this process. So mesosome formation and MREB tracks facilitate this partitioning of the chromosome into two daughter cells. One of the issues, though, is that DNA replication in rapidly growing cells is much shorter than the process, much longer than the process of cell division. So as I illustrate here, the cell cycle is completed in 20 minutes, but you need 40 minutes for DNA replication, whereas you only need 20 for septum formation and cytokinesis. And so how can this happen is the question that I have. And so the answer is that initiation of DNA replication has to happen much sooner. And it happens while these other things are starting as well. So there's multiple initiation points, and the timing has to be precise so that it precedes the initiation of cytokinesis. So on the x-axis here, you just see time in minutes for a typical E. coli cell. An initiation mass is reached, which means, all the things are built to, all the proteins and things that are needed for replication of DNA are produced. And then the origin of replication is, the replisome is assembled at the origin of replication. And then there's DNA replication starts. At this point, the bacterial cell has reached a threshold length where it's going to start dividing. But that's already here. You've already gone, all this stuff has happened already. And so that's how DNA replication catches up to cytokinesis. And so there's an initiation of the division process whereby certain proteins assemble at the division plane, and then the septum forms, the chromosomes start to be partitioned, and then you get septation and division into two daughter cells. So another thing that has to happen, if the cells are carrying plasmids, mind you, is plasmid segregation, where we know that plasmids replicate independently and they carry proteins necessary for segregation. So if these plasmids are important, the bacterial cell needs to retain them. So E. coli R1 plasmids, for example, produce 3 proteins that are essential for inheritance: par M, par R, and par C. Par M is similar to MREB in that it's an actin homolog and it forms long filaments. Par R is a repressor, and par C a centromere-like protein that both bind to the origins and link to par M, and I'll show you that. So par M filaments elongate and actually physically separate plasmids to opposite poles, as you see here. So here's a bacterial cell, and it's in the process of dividing this, replicating, and the plasmid is dividing so that the 2 daughter cells will each get a plasmid. And so here is the par M, the actin homolog filament par M, and then this is par R and par C together at the origin of replication here. And so again, these link the 2 plasmids together through the par M filament. And as the filament expands, polymerizes, it physically pushes the plasmids into opposite cells, and then this falls apart, and then each daughter cell has its own plasmid as well. This process isn't always this faithful. You don't always get 1 and 1, for example. There's some plasmids that replicate much more extensively, and as a result, it's harder to keep track of the 50 or so copies. So here we just see a plasmid that exists in single copy that's now divided into 2 copies and then pushed into 2 daughter cells. But again, you can imagine that some plasmids can exist in 50 copies or more. And so sometimes partitioning isn't so perfect. Cytokinesis or septation occur through these several steps. First, you get a selection site for the septum formation, and obviously that's an important thing to consider. The bacterium can't just willy-nilly just put septation enzymes anywhere along the length of the cell. It's got to be right in the middle so that you can get 2 equally sized daughter cells. There's an assembly of the Z ring, which is the actual zipper, essentially, that zips the 2 cells and cuts them in half. And I'm sorry, not cuts them in half, but it zips the elongated cell and cuts it in half so that it forms 2 normal-sized cells. But there is some kind of a anchoring thing that has to happen. So the Z ring has to anchor to the plasma membrane. And then, of course, it also stimulates the assembly of cell wall synthesizing machinery to repair anything at that site of septation. And then obviously constriction takes place. And so that, oops, here we are. And so the main players in this process are FTSZ, which is the main protein that forms the Z ring. It's a tubulin homolog, just like microtubules in a eukaryotic cell. There's a polymerization event that forms a Z ring, and then again the Z ring proteins are what almost like a purse string can pinch off the cell and divide it into 2 separate entities. This Min system, as I'll talk about, limits the Z ring to a certain location, particularly the center of the cell, and that's really important. As I said, you don't want formation of the Z ring to happen anywhere else but the center. So some of the other proteins in this particular case is this MREB. So I mentioned MREB is this kind of a coil that gives the cells shape, but it also forms tracks, almost like roller coaster tracks, where the chromosome can attach to, and as this is polymerizing, can push the chromosomes to the opposite poles, so that they can go into the daughter cells. But this process is also facilitated by the mesosome invagination, as I showed you. So some combination of those 2 events, MREB tracks and mesosome separation, both facilitate that faithful segregation of the chromosomes. So FTSZ, as I said, similar to eukaryotic tubulin, plays a role in Z ring formation. And so here's the Min system, again, the Min proteins forming this coil, and that the way this coil is set up allows the FTSZ to know where to set up as well. And so I'll show you that too. So again, just introducing you to the main players, FTSZ and MREB in particular. So this is a little bit of Z-ring formation, just showing the various stages. And so in this particular case, a receptor in the cytoplasmic membrane in conjunction with other proteins like ZIP-A potentially in the periplasm bind FTSZ. Once FTSZ is bound, that causes additional FTSZs to bind, and that forms that annular, that circular ring that will eventually pinch off the two cells and facilitate separation. By stage 3, a variety of other proteins with significant periplasmic domains are also on the scene, FTSA, FTSI, and then Q, L, W, N, K, this whole cluster of proteins are essential as well. And then when this all comes together, all the FTS proteins, including FTSZ forming its ring, in stage 4, you start to see the pinching off and septum formation. Another thing that's really important in this process are amidase type enzymes. And so amidases are enzymes that cleave amide bonds in peptidoglycan. And so here's the peptidoglycan chain up here, N-acetylmuramic acid, N-acetylglucosamine, and then the tetrapeptide side chain that's cross-linked to an adjacent tetrapeptide. And so the amidases will cleave here and remove this cross-linkage. And the reason for that, obviously, is so that the cells can septate. So in this particular case, you have a MZ is an enzyme that activates the amidases. And, I'm sorry, is a transcription factor that activates the enzymes. And in this particular case, actually, it's a it's an amidase activator, and that facilitates the cleavage. And so again, in order for these to physically separate, though, the peptidoglycan that keeps them together has to be broken down. And so the amidase's job is to break down the peptidoglycan so that there can be septation. If you have an amidase mutant, for example, that lacks the ability to break this down, then septation stalls. And as a result, you get cells that are linked together unnaturally. They wouldn't normally do that. And so amidases are really important for cytokinesis. And so this is just some microscopic evidence to show you that the central location of a Z ring. And so they used the antibodies to stain FTSZ green, and then the nucleoid itself here is stained with DAPI, a nucleic acid stain. And so again, you can see in these, this is just a cartoon representation of what's happening here, but there's the FTSZ ring right here stained with these antibodies. So clearly it's right in the middle, as you would expect it to be, so that this elongated cell can then chop in half and form two normal-sized daughter cells. So again, getting back to what I said, there's a couple different mechanisms in place to make sure that the FTSZ ring stays in the right place, in particular in the middle of an elongated cell. And so the MIN system facilitates that. There's another system in place, another mechanism in place called nucleoid occlusion. And so here's this cloud of genetic information. And so that cloud of genetic information prevents the FTSZ from setting up here as well. But the MIN system proteins basically can bind to the places you don't want the ring to form. So here on the poles, for example, you don't want a large cell. You don't want the cut to be here, for example, where you get a large daughter and a weird little malformed other daughter, and then this one's dead and this one dies too because it's too big. So the goal is for these MIN proteins, MIN-D and MIN-C, for example, to set up here, and then block FTSZ from ever being able to assemble here to cause cytokinesis to happen here. So between nucleoid occlusion, which blocks any ring formation here and here in this zone, and then the MIN system, which blocks any division sites here and here, the FTSZ proteins have nowhere else to go but here in the middle, which is exactly what the system is intended to do. And so again, it looks, kind of like this, where you have your origin of replication, your terminator, here's your DNA. The FTSZ proteins aren't able to set up because of nucleoid occlusion at this point, but because the cell is starting to elongate. At some point, the cloud of genetic information starts to pull to poles, and that creates this alleyway here for the FTSZ proteins to set up. Now the MIN system is in place and blocking FTSZ from polymerizing here and here. And again, nucleoid occlusion facilitates blockage until the chromosomes are pulled apart far enough so that the FTSZ system can form here, and then there's your ring that will form the site for cytokinesis. So cellular growth and determination of cell shape occurs through a variety of factors. And so penicillin binding proteins, which link peptidoglycan strands, they catalyze controlled degradation of new growth. And so these PBPs, they're called penicillin binding proteins. It's not really their job, but they tend to have that effect where, penicillin works by attacking these proteins, specifically transpeptidase type penicillin binding proteins. And as a result, penicillin can block cell wall synthesis and weaken cells and kill them. So they're called penicillin binding proteins because they happen to bind penicillin, but it's hard to believe that that was their evolutionary origin. Their goal is to, as I said, form strong peptidoglycan bonds. But again, in order for the bacteria to both build peptidoglycan and break it down when necessary to build new peptidoglycan, they need autolysins as well as enzymes that are more anabolic. And so this process is illustrated here, where the peptidoglycan monomers are being shunted from the cytoplasmic side to the periplasmic side using this carrier, bactoprenol. So UDP derivative of NAM and NAG are synthesized, so somewhere, and then in step 2, there's UDP NAM, and then there's a sequential addition of the pentapeptide side chain to the NAM. And then UDP NAM pentapeptide binds to bactoprenol phosphate, as you see here, and that relieves the UMP, and that provides us with this bactoprenol diphosphate NAM pentapeptide. So we call this molecule lipid A. Don't worry about this. This is just showing you where this compound cycloserine interferes with peptidoglycan synthesis and can be used essentially like an antimicrobial. But lipid 1 is the first thing that's built here. So pentapeptide, NAM, phosphate, phosphate. So one of the phosphates comes from here, the other phosphate comes from here. So that's where this phosphodiester bond is formed to bactoprenol. Then we add N-acetylglucosamine here, and so we end up with lipid 2, which is pentapeptide, NAM, NAG, and then the phosphodiester linkage to bactoprenol. Once we have lipid 2, which is the peptidoglycan monomer, the bactoprenol shunts this across the membrane to the periplasm, and so now this peptidoglycan monomer can now be added to the growing peptidoglycan chain. And so by this process, bactoprenol and lipid 2 facilitate adding new material to the peptidoglycan. And then there's just shown some places in which antibiotics interfere. So bacitracin, for example, interferes with bactoprenol so that bactoprenol can't properly bind here and can't properly form lipid 2 and therefore can't properly shuttle the peptidoglycan monomer to the periplasm. Vancomycin interferes with this cross-linkage step of new peptidoglycan monomers to the growing chain. And so as a result of that, these types of drugs, these glycopeptide type antibiotics like vancomycin and bacitracin interfere with peptidoglycan synthesis and can kill bacteria that way. And so again, this is just showing you the the autolysins in action. Here's the NAG UDP and NAM UDP, as I showed you, polymerizing into lipid 2. Here's bactoprenol binding to those and then dragging those into the periplasm so that peptidoglycan can be synthesized. But what this is really showing you is just some of the accessories, like the autolysins, for example. So in order for fresh peptidoglycan to be added to this chain, to physically open that up and add new material so that the cell can elongate prior to cytokinesis, the enzyme, these autolysin enzymes, have to come here and bust open these glycosidic linkages and break apart these bonds as well between the adjacent NAM peptide side chains. So again, obviously these have to be very controlled reactions, because you definitely don't want these things going haywire and destroying everything. So again, it looks kind of like this: the E. coli transpeptidation reaction and the Staph aureus transpeptidation reaction. So transpeptidation is a fancy term for, the addition of a new peptidoglycan monomer to an existing peptidoglycan framework. And so in this particular case, here's the side chain. Now an enzyme called transpeptidase will grab this here and link it to this here, and then kick the terminal alanine out so that you end up with this. Each chain of the peptidoglycan cross-linked to each other, so you get a nice tight bond and obviously a more rigid structure that can hold its shape and keep the bacterial cell together. So again, here's penicillin is essentially blocking this process too, just like vancomycin does. The transpeptidation reaction in Staph aureus, or gram-positives, so this is a generic overview for gram-negatives with E. coli as the model, and then here's a generic overview of the transpeptidation reaction in gram-positives with Staph aureus as the model. And so, as you can see, there's typically a glycine interbridge. So this is a direct linkage. This guy is physically touching this guy, but in gram-positives, this lysine is indirectly linked by a string of glycines to the alanine. And so transpeptidase will grab, this glycine chain that was polymerized in the cytoplasm and stick that right here and connect this lysine to this alanine, as you see here. And that's the Gly5 bridge. So one of the interesting aspects about peptidoglycan, it's also a toxin. We refer to it as tracheal cytotoxin, particularly with reference to Bordetella pertussis. And so this is again, the gram-negative model. Here's the LPS layer, the lipopolysaccharide layer, and then the inner leaflet, which is normal, a normal lipid. And so then you have some sort of a outer membrane transporter. And so, and here's the inner membrane, and then obviously the peptidoglycan layer. So all this is the periplasm then, when all this is happening. AMP-G is a permease that facilitates uptake of material into the cytoplasm, and AMP-D it removes the tripeptide, as you see here. So each of these little blocks is an amino acid on the N-acetylmuramic acid. So it starts here with lytic transglycosylase that cleaves chains into monomers. So it's breaking down. The constant job of lytic transglycosylase, basically acting as an autolysin, is to remove peptidoglycan monomers from the peptidoglycan so that the bacteria can grow. A carboxypeptidase, CP, that you see here, cleaves the terminal alanine off, and then in this form, the peptidoglycan monomer with the tripeptide can be transported by AMP-G permease into the cytoplasm, and then AMP-D breaks that part off, and then this thing can be recycled. So you can the microbe can eat this, metabolize it, or just build it into fresh peptidoglycan and pump it back out and put it back into the monomer. Put it back into the chain. So in this example, though, some of this material is pumped out of the cell, and then these peptidoglycan monomers actually behave as toxins. And so again, Bordetella pertussis, one of its primary activities is causing damage to the upper respiratory tract, and part of that damage is due to tracheal cytotoxin interfering with the ciliated epithelial cells of the respiratory tract. Lots of microbes use this, though. TCT is also used by Neisseria species, causing urogenital tract infections, and it's also used in by the beneficial bacterium Vibrio fischeri to remodel host tissue when it colonizes the interior light organ of the squid, which I'll talk about. So this is a little bit about cell wall growth and cell shape determination. So as I've already mentioned, the cocci divisosome, new peptidoglycan forms only at the central septum. FDSE determines the site of cell wall growth. FDSE may recruit penicillin binding proteins for synthesis of the septum. Rods are similar, but they elongate prior to septation. And so that's important. And in this particular case, so that the rod can be, so that the daughter cells can be a similar size to their parent cell. And again, MREB determines the cell diameter and elongation as Z-ring forms in the center. And so again, this is a little bit of that. So here's the divisosome, the septum that forms, and then basically once that happens, then you get hemisphere formation, and the cells elongate a little bit, and there's the two daughter cells. So this is all the new material, and this is the old material. With rod-shaped bacteria, you can see sidewall formation happening at various places along the length of the cell, and then the cell starts to elongate through a lot of different peptidoglycan divisions over here. But eventually, the FDSZ ring and new growth settles almost exclusively in the center. And then eventually, as I said, you get new growth, and then eventually you get septation. So there's some variations to the theme here, where again you have FDSZ that forms the Z ring. MREB is the helical polymerization protein that it facilitates chromosomal segregation and cell shape and so forth. But other cytoskeletal elements like crescentin can also occur in some microbes, and they cause certain bacteria to have a curved sidewall, for example, as illustrated here. So here's the MREB, and then this crescentin here that gives this guy the ability to be Vibrio shaped, a slightly curved rod. And again, the significance of this is who knows, but it may be to facilitate pretty rapid motility, just for aerodynamics. I'm not sure, but anyway, you see the juxtaposition of MREB and FDSZ to facilitating the way in which the bacteria would divide. And this shows you localization. Here's I think these are streptomyces, an organism like that has MBL, which is an MREP-like protein, and you can see it coiled inside the cell, giving this guy the shape, but it also, illustrates how this coiled protein can dictate how chromosomes segregate and where FDSZ aligns. Here's the crescentin in these cells, these Vibrio-shaped cells right here, giving them that curved rod appearance. And this is just FDSZ localizing to the center of cells to show where division needs to take place. So in this particular case, you're just looking at different kinds of microbes. So in these are the budding type microbes that will produce. So the parent cell here, they produce a little hypha, and then a bud comes off of that. And so just to give you a sense for, not everybody divides by binary fission and has the same issues. Streptomyces type organisms, they're bacteria, but they look like mold. And they germinate from a spore, but they produce vegetative hyphae. And then these vegetative hyphae make aerial hyphae, and then the aerial hyphae, just like molds, make spores. And so again, this is a completely different replication strategy in streptomyces, these bacteria, compared to E. coli. And I just put this in there. This is an actual electron micrograph that I took doing some work on fish gut microbes, and this is the intestinal epithelium. And then here is a microbe with a lot of slime on its surface, and then here you can see its hypha, and basically a bud will come off of that, and this guy will make a new daughter cell.